Today we will be talking about the role of the retinoic acid signaling pathway in lung organogenesis, so let's get started. There are 5 stages of lung development during human gestation. The first stage is the embryonic stage, which is during the 3rd to 8th week of gestation. During this stage, the lung bud forms. Also, the bronchial tree forms and starts branching, which continues into the second pseudoglandular stage, which is from the 7th to 17th week. The retinoic acid pathway mainly affects these first 2 stages. The third stage is the canalicular stage, which is where epithelial differentiation occurs from the 16th to 27th week. The fourth stage is the saccular stage, which is during the 24th to 38th week. During this stage the alveolar saccles form. The last stage is the alveolar stage, which is from the 38th week and continues into the first 2 years after birth. The mature alveoli form and proliferate during this stage. The retinoic acid pathway comes into play after the mother consumes foods containing vitamin A or retinol, such as carrots and egg yolks. In the blood, retinol travels with retinol-binding protein 4. Retinol can enter the cells of the splanchnic mesenchyme through either membrane transport with the stimulated by retinoic acid 6 membrane transporter, which is STRA6, or membrane diffusion, depending on gradient concentration. Once inside the mesoderm, retinol binds to cellular retinol-binding protein 1 or CRBP1. In the cells, retinol can either be converted into retinyl esters for cellular storage by lecithin retinol acyltransferase, or LRAT, or undergo enzymatic reactions to generate retinoic acid. In this case, retinol is converted into retinol by RDH10 or retinol dehydrogenase 10. Alcohol dehydrogenase is only used when retinol isn't binded to CRBP1. Retinol is then converted to retinoic acid by retinaldehyde dehydrogenase, or RALDH, 1, 2, and 3. Then excess retinoic acid is degraded by cytochrome P450, or CYP26. Some of the retinoic acid also aids in paracrine signaling. Most of the retinoic acid is transferred into the nucleus of the adjacent pleural mesothelial cells in the pleural mesenchyme and the endoderm-derived epithelial cells of the developing lungs, by binding to cellular retinoic acid binding protein 2, or CRAB-P2. In the nucleus of the mesothelial and epithelial cells, retinoic acid binds to the rare DNA binding site by binding to retinoic acid receptors, or RAR, which bind to retinoid X receptors, or RXR. This complex affects transcription of target genes, some of which will be discussed next. Retinoic acid inhibits or downregulates transcription of target genes like the TGF-beta genes, or transforming growth factor beta genes, such as TGF-beta 1. It also induces or upregulates HOX genes such as HOXA5. These steps activate T-box transcription factors such as TBX4 and TBX5, which activates the fibroblast growth factor signal, or FGF10. FGF10 activates FGFR2b, or fibroblast growth factor receptor 2b, which activates the NK2 homeobox 1 or NKX2.1. This leads to budding in the pleural mesenchyme of the mesoderm and the respiratory endoderm during the embryonic stage. FGFR2b also activates beta-catenin signaling, which transcribes BMP4 or bone morphogenic protein. BMP4 activates the SOX9 transcription factor or SRY-BOX9, which leads to expansion of the lung buds and branching morphogenesis during the pseudoglandular stage of lung development. As we mentioned previously, retinoic acid signaling plays a role in lung organogenesis as it impacts the first 2 stages of lung development, which are the embryonic and pseudoglandular stage. Retinoic acid does this by interacting with both the splanchnic and pleural mesenchymal layers of the respiratory mesoderm as well as the epithelial cells of the respiratory endoderm. These interactions cause and expand budding and branching morphogenesis in the lungs. Thank you.